So for now, it doesn't work. For this class,
I worked third shift last night.
for anatomy, but not for this. Not for this.
Right.
But like,
Apparently Johan's not coming. He might get
Okay.
No, I can't
Here's my
remember
notes
anyone
from
else.
last
Anyone.
oh no, these are anatomy.
That's why I moved over here. I was
Which I took the first day of anatomy, but now in anatomy, I'm gonna take my first class. But I didn't do that first. Because I take so much out of those those are my tactile.
You know what, I went to Starbucks this morning because it's right by my left. And then I got in my car and left it in there. Had to get more coffee downstairs. I was so mad.
I just put it on my first day.
Okay.
Yeah, I can do anything. In my car.
You're not missing this is what you already know. Like, IUDs.
And it felt so good yesterday. That was a 15.
I was surprised. I'm gonna do it.
I don't know if he's been
Yeah, I was like, wait, why?
Like, is it natural there?
I mean, if that's just,
I guess time around, not your period.
yeah, you're less fertile 14 days after your period or something.
But we know it's not.
How did you guys decide? Because you don't do it.
You're like, just like 14 days after your period, you're ovulating again. That's what she has for her birthday. We just, that's what it's, not what it says.
No, it's just like,
Three other people
basically.
say,
It
why are you here?
really helps woman like her body. It sounds something I'd say. Definitely.
Well, you're hospital.
No, just one girl.
No,
wait, what?
It's rather not.
I'd rather not take a shot in the dark on that one.
Right.
The years.
No, thank you.
Yeah, so we'd have to.
And
And
then
my
one person
ex-boyfriend, his
is in
sister was
charge
like,
of outline, and one
I don't
person.
know, like,
I was in and I wish that she had seen my
I've been doing this on a long time, so I feel like I'm being able to monitor all these things.
She was also one of those people that believed in the 2012,
you know,
breakdown of government.
Yeah.
So she never put her kids in school. They were in the wilderness.
And I'm like,
Oh my God.
Last time I talked to him, that was
my partner.
he said he hasn't heard from him, so they're living.
Like, I'll write 2 slides per, like, on like a board, with the information I want.
It was interesting though, so she's like,
Well, we have more time to do it.
my kids are like 5, 6, and 7.
Oh my God.
Yeah, they are. You know, you're just like, that stuff is real, them bills that
They are. We're on TV show on about the history channel.
Well, I mean, that seemed like this commercial. The funny part was, after the commercial, I saw a commercial for, it's just called the flu. And water to it is good for like 30 years or something. After a commercial doomsday preppers, like if you, it's like, it's dried or whatever, it's like good for 30 years or something. I'm like, I can't do that.
If there's no moisture in it, you can't really grow bacteria. That's a long time.
They go to tours free. My unit gave the whole friends for 2 minutes. They're warm, so.
Like I told him, like I got
It's doing good. David's our 6-month-old and Josh is our.
I have different, I have our lady and some have
There I is.
I guess not.
Oh, really? It goes back to his belly.
David.
Long enough for it. Jerry. He wants
First one was like 6'14". He was born like average size, but
Did you
What if like you was going outside and shovel steps to like one of the metal shovels and you fell down the steps and like the shovel punctured you or something? Or you like shovel and you fall like holy shit, you're like the stomach's like
So I get up and
well, I guess you'd be cold until you can bleed out. You'll just sit there.
That's okay.
Well, I guess I already cell phone is there.
That's how.
I wouldn't take them out. I mean, I guess you could just take it out. But I was just thinking, like, when it's going through, if they cut it, it's moving in your stomach, so you get hurt. Anyway, it's gonna hurt.
Right.
I remember this one time when I was younger, car. Well, they'll say, this is what we were driving, in the right lane, in the left lane. First the guy was going really fast to people, and then he just went around and like cut over, and then he overturned the flare. The car like twice. Of course, I was calling 911. and the other one. Like, and then the other one, like, I don't say that, but it was one lady stuff like that. They had to use that job to like take out the work, and she was alive, but they cut out the worst thing. She was saying like they was lifting her because of the pressure, you know. Like she was squished down. I guess she felt more squished down, but like when they pull it up, that was like, oh my god. So like her head was I mean, he just like, yeah, let's say he just hurried up and turned fast. You know what I mean? But let me see. Just everywhere. Like, it was like times two. I don't know. That was still. It was just, you know, people put their teeth on. That's what most of the thing was. They were saying, you want to hear the devices? Not going well. I feel like the attitude is much different or like much harder.
Right off. Are you alright? He's alright. I just wish he doesn't sleep a lot. He sleeps during the day. But not, I mean, at night it's better. Even though he's good, we're going to down the kids' time. Do we get the kids under like, no, maybe the devil never
I mean,
I used to, what do they do? Baths. I thought you got to keep that diet.
I guess that, I mean, you know, that's what I thought. No, of course you get to keep the driver's license. This one. Oh, I guess you don't want to like.
Now because they have murder games.
Do it yourself.
I love it.
No, I wouldn't do it. I wouldn't do it car. It didn't look like it.
It's like,
You know what I mean? Trying to go somewhere. Yeah, I'm an employee here. Yeah. Don't scan it. Just let me know. Yeah, she gets her accident.
Very deep.
We're ready to see those I think Patrick like gets to that. He gets
I like the bigger one. Did you get where did you buy bigger one? This bigger couch.
Which ones do you use?
Dr. Brown.
It's like a paper. Yeah.
So I
It's probably something I should have done today. I forgot about Bob. I really until like 2 weeks before he was born. knew that I was gonna be breastfeeding. I was like, yeah, but they
But I would also wait till the patient because I have the time.
like, what you not what your mom
They said it's supposed to be a negative 40.
Oh.
Negative 40. That sounds like some Arctic. I don't live in Alaska.
That's what this guy.
You know, I always wondered they could like drive snowmobiles to Wiggins.
That's why I'm
Or like, yeah, or like Target or something. Hey, I have to go get milk. They got the dog on the thing. And then they
Paul, why are you Snapchatting?
they have like they can take the Target cart home and hook it up to the snowmobile.
I just log in apps. Not very good.
I'm gonna go to carry the milk. They got the dog. You can pick off the one.
No, whenever he keeps talking about it, I'm like, I wish it did the great.
Here we are. Everybody made it. It's great. Testing, testing, testing. How we doing? Everybody okay? Anybody here today that wasn't here last week? You or Mindy? You were here last week. Oh, 2 weeks ago. I'm talking about 2 weeks ago. Wise ass. Okay, show and tell real quick. A lot of stuff going on in the field of microbiology. What do you got? What do you know? What have you heard?
People sick on a cruise ship.
Exactly, exactly. Evidently twice in the last 2 weeks. And sick from what? What's the bug?
Just the stomach bugs, all I heard.
That doesn't mean anything.
Was it like neurovirus?
That's exactly what it was. Norovirus, which is really short for Norwalk virus, but call it norovirus, and there are a couple that fall into that category. 300 and some people on the ship now that are sick. It's happened before out of this cruise line. So, what are the symptoms? Go for it.
Vomiting, diarrhea.
Yep. Severe gastroenteritis is what we would call that, which could be caused by 100 different organisms, bacteria and viruses, protozoans, things like that. So anyway, so that's going on. Anybody heard anything else? Go for it.
An increase in the H1N1?
Yeah, they're worried about that now because there have been, not a whole lot, but there have been enough cases of H1N1, which is what? Yeah, it's one of the flu bugs, and we'll talk about what those H's and N's mean as we move our way through the course. But it's a bug that, what do you remember about that? H1N1.
It killed a lot of people.
Yeah, and it was was one of those bugs that they were really, they thought it might cause major problems, and it kind of didn't. But one of the reasons it didn't was because so many people got vaccinated. And then a lot of people said, see, you were trying to scare us into thinking that this was going to be bad. And the reason it wasn't bad is that so many people followed the protocol and got themselves vaccinated for it. So that's another one that's out there. Anybody else got anything? I do. If I can find out what I did with it. This came off, where'd I get this one? Came off the microbiology, the ASM site, and it linked into a couple of other things, but I'll just read part. It's not very long. It says, once salmonella finds its way into a food processing plant, the bacteria can be extremely hard to eliminate. A new study finds researchers use 3 types of disinfectants to try to kill a buildup of salmonella bacteria. And the organism is growing what we now refer to as a biofilm. We'll talk more about that. And when you've got a biofilm, which you've all got right now on your teeth, especially if you didn't brush this morning, it's that film you feel, it's all these bacteria that are linking together, not just acting by themselves, but linking together in this film that's on there. And when there's power in numbers, is what it amounts to with the bacteria. The more you've got and the more they're stuck together, the more they're apt to not react to disinfectants, antibiotics, antimicrobials, whatever it happens to be. So anyway, that's the gist of the whole thing. Researchers used 3 types of disinfectants to try to kill up a build of a biofilm of salmonella that had formed on hard surfaces within the plant. And the study was published online, so and so on the Journal of Applied and Environmental Microbiology. In quotes, we found that it was not possible to kill the salmonella cells using any 3 of the common disinfectants if the biofilm was allowed to grow for more than 7 days before the disinfectant was applied. In other words, if the biofilm is already set up, you got a problem. And it's tough to clean this stuff. We found that all the types of salmonella we looked at were able to adopt the specialized biofilm lifestyle, and all the surfaces we looked at, including glass, stainless steel, glazed tile, and plastic. The biofilm of salmonella gets more dense over time and becomes even more firmly attached to the surface. Corcoran said that the findings showed that food processing facilities must keep salmonella out of their clean areas where cooked food are further processed and packaged. People need to question whether disinfectants that are promoted as killing various types of bacteria are really as effective in real life situations where biofilms can form so and so and so and so. And it goes into make applications into your home, and that's really the point of the whole thing, is that you see in your bottle of spray something, you got Lysol, it says kills 99.9% of the bacteria. That's under lab situations looking at colonies which have, on a petri dish, which have not formed a biofilm. So once a biofilm is set up, whether it's on the, your Tupperware, the inside of your Tupperware, you ever wonder why sometimes you get food that's in the refrigerator for weeks on time, and you find it in the back, and then you take it out and you wash it, and it still feels slick? That's why. Because that biofilm is set there, and then you wonder why food rots in there the next time you put it in there, or wonder why you keep getting sick. Word to the wise about that. Just because it says something on the bottle like that doesn't necessarily mean that it's true, which takes me back to another adage which I told you we're going to learn in this course, is to trust no one. Okay? Look around the room. Trust no one. And to be a good skeptic, when you hear stuff, so on and so on. I was with a guy yesterday afternoon. We were involved with putting together a snowblower or something like that. And he was going on this rampage about people just don't drink enough water. And if you drink this amount of water, this is what happens to you, and so on and so on. And so which I always do, is that, how do you know that? Well, everybody knows that. So what's the adage about water? Okay, this came off the NPR site just a couple of years ago. Myth number 1, drink how many glasses of water a day? Okay, you cannot find the research that shows there's any validity in 8 glasses of water a day. So, as they say, scientists say there's no clear health benefit from chugging or even sipping water all day long. So where does the advice of drinking 8 glasses come from? Nobody really knows, says Stanley Goldfarb of University of Pennsylvania, who's a kidney expert. And yet this is one of those things that kind of enters into our conversation, and we just assume it's true. You know, God sent this down on tablets. Drink 8 glasses of water a day. There is no research to prove that somehow is this magical, mystical, important number of glasses you've got to drink. Tell your nursing faculty that. Myth number 2, drinking lots of water helps clear out all those toxins. That's what this guy's going. You need to clear your toxins out of your system. Myth. The kidneys filter your toxins out of your bloodstream no matter what. Then the toxins clear their way through the urine. The question is, does drinking extra water during the day improve the function of the kidneys? The answer to that is no. So the idea that, well, I'll drink more water, that helps my kidneys flush crap out. Wrong. No research to support that, okay? In fact, Goldfarb says if you drink 8 or more glasses of water a day, it really reduces the kidney's ability to function as a filter. Think about that. Myth number 3, lots of water equals healthier skin. Oh, sure, excuse me. So I won't read what he says in there, but he said there's, again, no research to show, but Procter & Gamble and Unilever report this all the time because they put out all these skin products. But there is no research to show that's the case. Drinking extra water will lead to your weight loss. A more accurate statement might be drinking water is a helpful tool for dieters. See the difference in the approach? One saying that extra water is going to provide weight loss, but the real statement should be that drinking water may be helpful for people who are dieting. And for what reason? It gives you a sense of fullness all the time in the stomach. Yeah.
If you're drinking water instead of soda, you're hundreds of calories a day.
You got it. Exactly. Myth number 5, and I won't go on with this forever. It's easy to get dehydrated during a normal workout. Okay. Dehydration sets in when a person has lost 2% of his or her body weight. So a 200-pound man, this means losing 4 pounds of water. A marathon runner, bikers, and hikers all need to recognize the signs of dehydration, but it's obvious that individuals in hot, dry climates have an increased need for water. American College of Sports Medicine recommends that an athlete drink 12 ounces every couple of hours before starting practice, so on and so on. Goldfarb's advice with all of this is only drink when you're thirsty. Okay. So anyway, things to keep in mind. Don't believe everything you hear. Okay, where are we with this? What did we do last time? It seems like years ago when we were together. But we spent some time talking about kind of major historical events in the field of microbiology. And so what were those? We talked about the invention of the microscope. We talked about von Leeuwenhoek. We talked about Hooke being important in that. Then we talked about this discussion over abiogenesis or biogenesis. Abiogenesis, another term we gave to that was spontaneous generation. And then we talked about the fact that with biogenesis it means life comes from life. And with the use of the microscope, individuals began to see organisms reproducing, and it supported that theory. And then we talked a little bit about what Pasteur did with that, trying to use his swan neck flasks to say that you've got an open system, but as long as you can't let the bugs in, you're not going to end up with contamination of the media you put in there. And so he tried to debunk that theory of spontaneous generation with his swan neck flasks out of all of that. And then we talked about the germ theory of disease, which kind of emanated from all of that. And we talked about Koch and Pasteur both being involved in that germ theory of disease, developing that, along with a lot of other people. So we're not mentioning all these names, but we're trying to throw a couple of important ones out there. And then with the germ theory of disease associating an organism with an infection, we're talking about infectious disease, quite obviously, as opposed to not every disease, but with infectious disease. And we talked about how that really started to come together, and we made some sense out of that in the latter part of the 1800s. So it's not that long ago that we started to see this idea that this organism that you can't see is the one that's actually causing this disease and making you feel this way, et cetera. With that came the golden age of microbiology. We kind of talked about that, which ran from about 1855 ballpark, 1855, 1860 to about 1912. And it was during that period of time that not just organisms were being discovered that cause disease. Koch discovers the tubercle bacillus that causes tuberculosis. Anthrax, Pasteur also discovers the organism that causes anthrax. So we're associating organism with disease and all that kind of stuff. But then the methodology of studying these organisms is going on at this point as well. So we're learning how to make microbiological media so we can grow these organisms in the lab. We start to come up with the tools, petri dishes, so on, staining procedures. Most of that procedural stuff came out of whose lab? Koch's in Germany. By far, a majority of it did. The whole idea of using agar as a solidifying agent, we'll talk a little bit about that in the lab this week. So we have that kind of an event going on, and then we said, okay, so we've got that organism with disease, and maybe the next step would be to say what? Let's keep those organisms out. And so that led us into the whole idea of disinfection or antisepsis. So how do we control these organisms out in the environment? And we talked about who? Lister. And the fact that Lister used phenol or carbolic acid in his surgical arena to reduce the post-surgical infections, and it worked quite well. Ended up killing him. Got to hold that whole thing, and then we said, well, what happens if the organisms get inside you? And then we talked about the whole idea of not antisepsis, but antimicrobials, chemotherapy, antimicrobials. And we talked about the development of penicillin, quite by accident with Fleming, the idiot. And Florian Chain, who really were the guys who did the main work with that. We talked about, who else did we mention there? The development of the sulfa drugs, Domac. When did all this happen? 1920s and 30s into the 40s, and then we step into streptomycin, it just goes on and on and on. So by the 1950s and 60s, there's just one antibiotic after another being discovered. And you know, what's happened in a short period of time? Going from the 1960s to where we are now, in 50 years, we've squandered that wonderful use of something that is very helpful. We've squandered it badly, and now the bugs are laughing at us, saying that it doesn't work anymore. Then we said, okay, if we can kill off the bugs selectively that are in you, that whole idea of, and even salvarsan with air, like we throw in there with that whole thing, can we go with it?
I have a question.
Sure.
Was there a reason to suspect the development of superbugs from, at that point in time, a long time ago? The problem is we've given out so many antibiotics for every little thing.
Yep. Mostly for animal feed.
And so that's helped produce some of these antibiotic-resistant bacteria.
Exactly.
Would there have been a reason to suspect that?
It's a natural process, quite honestly. So that the bacteria out in the soil are going to be exposed to antimicrobials that other bacteria produce or other fungi produce. It happens all the time. And there are going to be certain bacteria in the soil that get exposed to that. They're going to be killed off by that stuff. And there are others that are going to be resistant purely by accident because they've got a mutated gene that allows them to produce this, that, or whatever, protect themselves. So it's a natural process. All we've done is push the process forward by dumping millions of tons of those antimicrobials into the environment. and selecting for all of those organisms which are now resistant. So we didn't cause the resistance, it's a natural process. We've simply said we can increase the number of bugs which are now resistant, which we do in the hospital, right? Somebody comes in with an infection, what do? We treat. In fact, we treat probably half of the people in this hospital at any one given time with some type of some type of antimicrobial, not always antibiotics, but antimicrobials out of this whole thing. And what are we doing? It's a sealed environment, and we're only killing off what? The weak ones. So what do we keep concentrating in the hospital situation? Are those resistant bacteria. And that's why probably a third of you right now are positive for MRSA. You're not sick, but you're simply carriers because you picked it up by walking through the halls of this hospital. And then you give it to a patient and kill them, but you don't know that because they're gone and you're somewhere else. But we'll talk a whole lot more about that as we move through. Then we went with what? I'm trying to think. Let's develop systems for immunizing you. So that you're going to be exposed to bugs, that we can work up your immune system to the point that when the bug shows up that you're ready to deal with it. And so who was involved with that? We talked a little bit about what? Jenner, I think, we mentioned him before the golden age of microbiology, but what did he see? That relationship between milkmaids who got cowpox, the fact they didn't get smallpox, and they developed some kind of resistance. He didn't know exactly what was happening, but he just saw that the system worked. And last of all, we may mention the fact that now with the microbes, where are we with these? Well, we realize that we can manipulate them genetically, and we've learned how to undergo that whole process of genetic engineering using them, and we've made those applications beyond the microbes into crops, into animals, and into humans. So genetic manipulation, genetic engineering, and so on and so on. So those are some kind of major events that we kind of talked about. There are a whole bunch more, but we just don't have the time to deal with all of them. But we'll kind of build on these as we move our way through the course. Okay. So that's where we were last time. Let's jump from there. I gotta reach. Barely. What's that?
Oh, I was wondering,
Oh dear. Go ahead.
so you know how everybody gets vaccines and everything like that?
Yeah.
Why is it some people now aren't getting their kids vaccinated? Why?
You have as good an answer to that as I do.
Oh.
We go through cycles where parents do not want to have their children vaccinated for all kinds of reasons. Some of them feel that there's still mercury-based vaccines that are causing everything from autism to this, that, or whatever. And we've debunked that whole thing, although that still shows up from time to time on television. And it's usually word of mouth stuff. Somebody says, don't let your kids be vaccinated because when my kid got vaccinated, this happened. They got the flu. And it's by word of mouth stuff. And so parents kind of get together and go, some lady down the street told me this. And that should never be your source of information, right? Okay? And that's why even looking at each other, half the people in this room are morons. Don't trust them. Don't trust them. Well, I'm not saying who's who, but don't trust them. But that's basically where it comes from. It's bad information that gets spread from person to person. So the adage is, trust no one. Okay?
Does that apply to you as well?
Absolutely.
Right, how do we know what you're telling us to do?
You can't trust me. You'd be a fool. Oh.
Yeah, you might leave us in the dark.
Exactly. Or lead you places you don't want to go. And you don't know whether I'm just making this crap up, do you? No, you don't. You don't. Okay, take a peek at this. You got something very similar in your textbook, but let's just talk a little bit about this. We need to spend a little bit of time with the idea of classification, and I'm not going to beat this into you, because most of you don't find it really interesting, but there's some concepts that you need to kind of get under your belt as we move our way through the course. Well, that thing is filthy. So if you take a look up there, you see in these purple boxes, from working from an angle, it's not the easiest to see. Is that purple or violet? Purple. So we see some words we know, like animals, and plants, and fungi, and protists, and monarans. This is an attempt to kind of say, how do we classify all these living things on the earth? And the typical approach was prior to about the 1960s doesn't go back that far, was to say that everything is really kind of plant or animal. And it was really that simple until a guy by the name of Whittaker, see his name down at the bottom? Whittaker. Said it's more complex than that. And one of the reasons why this was difficult to come up with this is because we really didn't have a good way of studying genetics in these organisms. This is still early genetic study time, even though we're just starting to put pieces together. This is only 10 years after Watson and Crick have kind of done their thing. So this is all relatively new. But Whittaker said, instead of just plants and animals, we really have a variety of different groups here that we have to separate from one another. And we've got some areas over here where we kind of don't know what to do with these. But he said there are protists, which are basically these things that von Leeuwenhoek called animalcules. And those are what we now call what? Be more specific. Protozoans, exactly right. And so he used the term protists up there, which sometimes is still used. And of course we got the animals and we got the plants, but the fungi now get stuck in there because prior to this time the fungi were dumped in with the plants. Well, it couldn't be farther from the truth. Fungi don't photosynthesize. So why put them in with organisms that do? But so Whittaker says, let's give them their own little group up here. And then he said, I really don't know where to go with these kinds of things. These are algae. Are they plants? Are they derivatives of plants? And so this was kind of a question mark. But the other thing he said was, you notice this dotted line running across this, that we're going to put all of these into a group called the eukaryotes, in which U, in this case, EU, I don't know if you can see that up there, it's sometimes spelled with a K. He's got EUCARYOTES, the eukaryotes, and U as a prefix implies what? True. The karyos in this case refers to kernel or nucleus. So we've got organisms, everything above this dotted line that you see here is eukaryotic, meaning that these cells have a nucleus in them. Now, are there some exceptions? What about? Okay, so give me an example in your body where we might argue. Red blood cells. So as a mature cell, a keratinous or a reticulocyte or an erythrocyte is not nucleated, correct? But it was at one time. It simply specialized and said, I don't need this nucleus anymore. I've made all this hemoglobin. Out it goes. But we're saying that these are organisms that have that true nucleus. And by true nucleus, we're talking about a nucleus with a nuclear membrane, all those chromosomes, that genetic material inside that. And then he used a term which is rarely used today for the bacteria and bacteria-like organisms, and he called them at that point the Monera. So if you look at all of this, what you see is that Whitaker came up with something referred to then as a 5-kingdom approach, in which all these these purple boxes represent a kingdom. So the Monarans, the Protistas, the animals, the fungi, and the plants. But then now we're starting to use some other terms here, saying that we can take these 5 kingdoms, 4 of them fall in this group called the eukaryotes, but the Monarans fall in this group called the prokaryotes. And the term prokaryote, which is in bacteriological circles right now, it's a huge argument that's going on, saying we shouldn't even use that term anymore. And little by little, they're trying to phase out the use of that. Because pro indicates that the bacteria gave rise to the eukaryotes. And with all the genetic studies, we can't really support that well anymore. So that's an argument you don't have to worry about, but it's going on right now. So everything below the dotted line, are what we're referring to then as the prokaryotes. Prokaryotic organisms. You hear that term a lot. And basically those are the bacteria. So we live with that, Whitaker's approach, this 5-kingdom approach, until we start to work our way into the 1990s. And then somebody shows up, Carl Woese. Can you spell Carl's last name? Excellent. W-O-E-S-E. Woese. That's damn close. Carl Woese. W-O-E-S-E. And Carl, working out of a university in the Midwest, virtually spends his life trying to make sense out of all of this. This tree of life kind of thing that who's related to who and so on and so on. And he ends up studying ribosomal RNA. Ooh, everybody have a clue what that means?
It's inherited from the father, the male.
Okay. And its function? It's protein synthesis, site for protein synthesis. And to keep the story short so I don't lose you, basically what he says is that looking at that type of RNA, that all these organisms fall into one of 3 groups. You either have RNA, excuse me, and all of these organisms that Whitaker divided up into kingdoms, these are all basically the same. Looking at that RNA, they have the same RNA. If you look down at the Monarans, though, he said, ooh, I've got 2 other groups down here. Here's the bacteria, but they were already down here, and they in turn are going to get their own kingdom level at this point, which he then said, I need to change that name, and I'll come to that in a second. But then he said there's this other group that was discovered not very long ago at all, over the past 30 years, called the Archaea. Archaea says what? What does that sound like? Archaeology. Archaeology is the study of things that are old. So the idea was that now we've got this one group of bacteria, and it's the Archaeobacteria, but everybody just shortens it to the Archaea, who are bacteria-like but have their own type of ribosomal RNA, and therefore we're just going to put them in a separate group. The point of the whole thing is this. Woese, Carl Woese, says there's another way of looking at this. And if we look at it from my way, looking at ribosomal RNA, there are 3 domains. He said, keep Whitaker's system if it works for you. But let's overlay the fact who's related to whom. So we've got this domain called eukarya. That's in the golden box. This is Woese stuff. Everything above is very closely related, which means that if we see ourselves over here among the animals, we aren't that far off from the brown algae, marine algae out there, based on this. And he said then we've got the bacteria, common bacteria that we know. We're just going to call those the bacteria, or maybe the eubacteria, sometimes that's terms used. And the 3rd domain of the Archaea, which seem to be kind of odd and maybe old. And now we look at that and we're saying, That's probably not the best term to use because it implied that it was the oldest of the bacteria. And it may not be true. So here's the gist. Whitaker, how many kingdoms? 5. And you remember how kingdoms are set up? Kingdoms, all that stuff, all the way down to genus and species. Woese says, let's generalize even more based on genetic information, and there are 3 domains. 3 domains. Okay, so what we're going to function with today is focusing right here. Everybody with me? So we're going to look at these prokaryotic organisms, that group that are referred to as the bacteria or the true bacteria. And we're going to spend most of our course dealing with that particular group. Everybody with me? You know, if you want me to repeat this, I will. I mean, I don't want you to get lost along the way. And many of you are going, what does this have to do with nursing? It doesn't have anything to do with nursing. Or broadening your horizons so that you're an educated person. So that after you've killed the patient, you can talk about something else. Okay, everybody's there? Okay, let's roll. What should we do first? Let's just throw this up. It's not a good overhead, but it's so stupid that I like it. Okay. Photomicrograph of a bacteria at the top. So this is done with the electron microscope. We're probably up to several 100,000 times magnification. Leeuwenhoek said what he found were these things that he didn't know were bacterium yet. These little critters that he kept finding over and over. Some of them were round, remember that? Some of them were curved and some of them were rod-shaped. Well, this would be one of those that's rod-shaped. And if you take a look at it, you kind of go, okay, so it's magnified several 100,000 times. I've got something here, right? I got something there and there. But other than that, everything just looks kind of granular. So what do we got here? Well, if we draw that picture, taken from the photomicrograph, and we simply pencil it in or ink it in, whatever it is, what we see is that we've got, working our way from the outside, is that we got a cell wall, not a cell membrane, a cell wall. Inside that, there is a thin cell membrane, which the cytoplasmic membrane or the cell membrane inside of that. We've got this cytoplasm. That sounds familiar, correct? And in that cytoplasm, a phenomenal number of ribosomes. Phenomenal number. That's what gives it this kind of granular appearance. And when you see excessive numbers of ribosomes, it indicates what? Yeah, we're making a boatload of proteins. And remembering again that those proteins can fall into one of 2 categories, ANP1, lecture number 3. Those proteins can be what? Used for structural components in the cell, whether that's actin or myosin or something like that. Or they can be functional. In your case, or in my case, like hemoglobin, or maybe what else? Enzymes, exactly right, which are functional proteins. So it indicates that this cell is probably very active metabolically. Bless you. Did somebody use proper respiratory etiquette?
I don't know.
Did you use proper respiratory etiquette? Oh, excellent. But then we look here, and what do we have? It's not a nucleus, but it's called a nuclear area. Again, that's quite all right. Well, good. A nucleoid, sometimes called that, or a nuclear region. And what makes it different is that from a nucleus is that it doesn't have a nuclear membrane. There's no membrane-bound nucleus here. In fact, if you start to look you know, all of those organelles, those membrane-bound organelles you had to learn in A&P, and give me some examples. Nucleus, obviously, Golgi apparatus, endoplasmic reticulum, exactly, and so on and so on and so on. Do you see any of that crap up here? No, no. So the only membrane-bound structure you see here is what? Cell or cytoplasmic membrane. That's the only membrane we see out of this. And yet these cells are incredibly active metabolically. So, and mitochondria? No. So what should be going through your head right now is, how do I make sense out of any of this? How can this cell be that metabolically active, can do all of these things, reproduce incredibly fast, in some cases every 15 to 16 minutes if everything is right, and you wonder why, when you got that slightly E. coli tainted hamburger, undercooked, you wonder why in 8 hours later, you know, you have uncontrollable diarrhea. And what's happened is that organism has reproduced very rapidly in a short period of time. How can it do that? Obviously, to reproduce, you need energy, and lots of it. Lots and lots of energy. Where's that coming from? These are the questions that ought to be going through your head right now. And yet these organisms are incredibly, what? What words might you use to describe them? They can kind of live anywhere, right? They can reproduce rapidly. They can metabolize things very quickly. So what they're carrying around is minimum baggage, but able to get things done more rapidly than we can. So the term that's sometimes used with these, when you talk about the bacteria, these little guys, is that these are the simple cells. Anything, I mean, that couldn't be farther from the truth. They're not simple at all. They just can do things better than we can with less stuff. And the question is, as we work our way through the course, how do they do that? Okay, we got these dense things here. This is not typical of every bacterial cell, but it's just trying to indicate that from time to time certain species of bacteria may store things. Could be iron, could be lipids, could be phosphates, could be a whole variety of things depending upon what the organism is, the species is. But whatever is stored is never stored in a vacuole. Because a vacuole has what? A membrane. But we're not doing that here. So if we take a look, this is just the goofiest picture because it always reminds me a little bit of like a bowl with pasta in the middle. But if we were to take that cell and slice it open, we've got this nuclear region, as it were, with its DNA in there that kind of looks like tossed pasta, and then we've got this cytoplasm here with many, many ribosomes. And are ribosomes membrane bound? Say no.
No.
No, thank you. They're what? They're particulate organelles. They're non-membranous organelles out of all of that. Okay, so let's make some sense out of this whole thing. Where do we start? Well, one of the things we see is no matter what kind of bacterium we're dealing with some exceptions, that around the outside of this, there's going to be a cell wall. I have 1,000 handouts here. I do. No, there. Handout coming out. Stop yawning. Run some that way. Everybody's car start this morning? Excellent. How we doing?
Could you bring these in a better focus or something? Because I don't know, they're really hard to see. I'm not that far away. Is anybody else who just?
Is it hard?
Yeah. Okay.
But now it's.
A little better? That's everybody. Thank you very much. Is that better? I know. Again, photomicrograph, taken with the electron microscope. So we're magnifying hundreds of thousands of times on the edge of that cell that we just saw before. And here's where we see that cell membrane, that little thin line down there. And again, these are so small that you're not going to get a highly defined picture, although we can do better these days. But outside of that, there's some word out here, peptidoglycan. We've got to make some sense out of that. So what we're going to look at right now are cell walls, cell wall structure, because bacteria tend to fall into one of two categories based on cell wall structure. So when you hear the terms gram-positive and gram-negative, you've all heard those terms. What does that mean? Where does that come from? Well, we know that certain antimicrobials are going to work against gram-positives and not against gram-negatives. Some of them work on either of those, etc. So let's make some sense out of this gram-positive, gram-negative stuff. And what it boils down to is cell wall chemistry. So we're going to take a look at this outer layer, that word peptidoglycan, and we're going to make some sense out of the handout that I just gave you with various ways of trying to depict this stuff. And this is stuff that I stole from other books along the way. But let's start up here. That looks out of focus to me. So here's the cell wall, excuse me, the cell membrane. Do not confuse the term membrane and wall. Two entirely different structures. So here's the cell membrane, and you recognize that by looking at it when you see that kind of configuration that's up there, because it's what? Biphospholipid membrane. It's got that unique kind of structure that we always draw with what? Yeah, and what are these things that are out here? Those are, what is that molecule? 3 carbons? Triglyceride. It's a glycerol molecule. That's what the globular end is. And what's hanging down? Fatty acid chains. And we call these phospholipids because instead of it being a triglyceride, one of the carbons is gone. We've added a phosphate group onto that, so it's a phospholipid. And what do we know? Water. Exactly. Hydrophilic to the outside, hydrophobic to the inside. So that structure you know already. You've heard that numerous times. But then to the outside of that, we start to see this unique kind of stuff and how it's made, which makes up what we call the cell wall. And the term that's used here is something called peptidoglycan. Take the word apart. Peptido sounds like peptides, sounds like polypeptides, sounds like proteins. Glycan, anytime you see the prefix GLY, you know exactly what we're talking about. Sugars. So it's a mixture of, in this case, amino acids and sugars. That's what that's what peptidoglycan is. But let's get more specific with that. If we take a look at the part of this chain, which is the sugary chain, the 2 sugars that are there are here, N-acetylmuramic acid and N-acetylglucosamine. Now, you probably have a tough time with that, but look at the handout. And look at, what should we look at that's easiest? Let's look at this side. Again, these are taken from 3 or 4 different textbooks. And I just kind of stuck them all on here so that you can come up with some kind of an idea. And look under A on this side, which is to your left. Everybody there? And you see the GMs? GM, 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 GM. Yellow? Okay. The G stands for glucosamine, the M stands for muramic acid. So if we use GM, 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 maybe it's easier for you. And what do you notice is that on A, there are these long chains of sugars, and then you see those little dots. Those little dots are amino acid chains. Short chain proteins, they're short chain polypeptides, call them what you will, but there are these little links that hold them together. And on the diagram, they're represented by these dots. On this attempt up here, the blue rods going back and forth are the sugars chains, and in this case, the golden rods coming across are the short chain amino acids. And what do you notice? Multi-layered, very organized. Everybody see that? Absolutely. This is typical then of what we would refer to as a gram-positive bacteria's cell wall. This is what it looks like. So the A on that part of the handout and what you see up here is typical of a gram-positive organism. Multiple layers of peptidoglycan bonded or held together with a very organized set of short-chain polypeptides. Short-chain amino acids, call it what, use the language that makes it easy for you, because we can use a variety of words. So it's thick, it's strong, it's organized, it's regular.
Can you repeat that? I'm sorry.
It's thick, it's strong, it's regular.
What you said, gram-positive is made up of what?
Gram-positive is made up of multiple layers of these sugars. In this case, you see what, 1, 2, 3, 4 layers? If you were to flip your handout over, look at the bottom of the other side, and this is trying to show a 3D effect, and it maybe didn't work quite as well. But you see what they're referring to as this carbohydrate backbone? That simply means these sugars. And there are these long chains, GM, 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 one after another, and they in turn are linked together with these tetra peptide side chains, these short chain amino acid bridges, whatever you want to call them, that hold the sugars together. It's thick, it's regular, and it's strong. Typical gram-positive organism.
So is the figure 410, is that a gram-positive also?
On 410, exactly. Now look at the above on 411 on your handout. A. And there you see the plasma membrane again. It's simply another way of somebody drawing it differently. That's all that this is. So instead of making the sugars as circles, they're as little rectangles. And you could write GM, GM, GM in there if you wanted to do that. And you see these slanted lines coming across? Those simply represent those short chain amino acids that are holding all of this together. There is one other factor here, though, that you notice on this particular drawing, though, and it says something about teichoic acids. See that? It's on the top left of your handout. Teichoic acid. It looks like a thread running through the cell wall. Everybody with me? Say no if you don't.
Yeah. No. Yes.
Okay.
Yes.
No, yes?
Yes. Now yes. Yeah.
Now, yes, now, no, now.
Yes.
Yes. And teichoic acid, we'll talk more about those later because those sometimes become what we call a virulence factor. Any clue what that might mean?
No.
If something is virulent, yeah, it has something to do with it's a disease causing something. And we're going to find that one of the things about bacteria is that they have a variety of virulence factors, and teichoic acid can be one of those virulence factors that helps it cause disease. Yes?
The short chain amino acids, are those the tetrapeptides?
that hold together the
Those are those,
sugars?
yes. The whole mess up here is called peptidoglycan because it's a mixture of sugars and proteins. And it's a lot of words that overlap each other, and I understand why there's some confusion. So we can say we've got a peptidoglycan layer in this gram-positive bacteria. Here's the peptidoglycan layer of the cell wall. Now let's look more specifically at it. What's the glycan part? Glucosamine, uramic acid. Those are sugars. They line up in chains. GM, 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 long chains. The chains are held together by these cross bridges of short chain amino acids. So it's strong, it's exceptionally strong out of all of this. And while you see 4 layers of this peptidoglycan up here, some bacteria have many more than that. Yes?
So the gram-positive organisms, will they always have the GM thing?
They will. They will, they will, they will. Now, I should never say always and never say never. In the sciences, you never do that. Let's just say that it is the typical formation that we would see with gram-positive. Are there variations on the theme? Absolutely. But we're not going to go with the variations right now. Let's keep this as simple as we can. So this would be typical gram-positive cell wall chemical structure. Let's take a break. 15, I'll meet you back in. Take 15. I'll see you back here. Ask it. This is peptidoglycan. These are the layers. This would be.
Oh, it's not.
All the way up. Those chains represent this chain or this chain. Glucose. The M stands for uramic acid. Say that. Work on it. Close. Sure.
Yeah.
Feel free. Give me the white paper. Now, in your textbook, there's a picture that's almost identical to that. Almost identical.
And he's doing like everything so. I'm a faculty, so it's like crack your head eyes. It can make my kid cry.
I guess that's why I'm like, I hope it wasn't. Have you seen that quiz or something?
It's got some accuracy.
Have you seen that thing? I should have
This time last year. I can't get to butt, like I'm just feel like I'm just gonna tired.
I think it's, I don't know why, I feel like I'm mad, but I gotta be in it. I gotta be in it.
And I was trying to impress myself when I got
Me
there.
too. I like my teacher. Which one is that? He seems like he's pretty good. I just gotta keep up with him. And I'll let you get to me too, that's what I'm saying. Just ignore it. It's like it's in the book and then he say, then like whatever comment. And then he told us not to read it. Remember, he's like, don't read it. Read the bold, like the bold. So I'm like, let me read it. Oh, my brain.
It might be the same. Maybe like his own comment.
I guess the book, after our class, we can go through the chapter, you know what I mean? After we know what he focused on talking
Now, this kid I'm talking to, he's taking micro-life pieces in there right now. And you see him just like,
Really?
she loves it. I wish I was there.
I really like.
I'm glad I'm here. I just wish I would have transferred.
I know, right? Oh my gosh. Like, I had to take this on because I had to retake.
Yeah, I like to say.
Like,
I
my own
ninth grade. We had to make our own project. I made like reading plants.
And then he's like, what is
It was like a picture of each paper. And I came in like,
yeah. Right? Oh,
So
yeah.
that means I took A&P and like,
I took it last year.
See, I must have got it fresh out. I was like, I got my AD book like right there. And I'm like, I need to be.
It's really booking it so back with my
Yeah.
I kept like,
I can't forget.
by like magic.
Yeah. Like, when I was they just say something nuts because I was
Unless we all mutate. I know I have it.
I think
hopefully he'll give us like, and you see how, where he went over everything. I wrote like my notes. He went over, you know, he numbered it. It's like he doesn't remember what we should have. You know how he numbered it in the chalkboard. And he reviewed it. So hopefully he'll probably do it. Like a review of what we should have.
That goes 7 miles.
My girl, Chem 1. Tomorrow I have micro lab and lifespan development. Chem lab.
going down my street. See, I don't drink coffee.
But is yours more?
Mine's on Wednesday.
So I love it. I'm like, come here.
Oh, you're in the field, right?
Already plowed with top plow. I don't know why. But he was just tearing it up like
It's all right, we got it.
I mean, it was you know what? What are your boys' names? Oh,
You don't seem like he does. So I'm like, this is my goal for it, and it shouldn't even be that bad, because it should be like a review. I don't know how you teach like that. That's the first time I'm looking on this, so I teach that first. Oh, it is, so you guys journals and stuff yet? I know.
Right now I call him baby. Baby. But I don't know what we're gonna call him. Jason had this shit. I just think it's only for it the other day. It sounds like Gopher. I'm not sure if I'm a fan of it, but it's
You're really serious.
but I don't know if I will like it.
I feel like that's
really
Gopher Grace.
You get extra points on your certification. competition doing
And that's the only one I can think of.
It's what do you get extra? What do you get extra points for doing?
Oh, so everyone is talking.
Like it wasn't like a
So
PowerPoint.
That's what we're doing. Whoa.
record his lectures and read
Like, you guys
his
just-
lecture in his,
No, well, we did.
you know, the other part of the lab. Lab lectures, record his lab lectures.
I was like, what? I didn't know there was another part.
Yeah, we have
Everyone said they recorded those.
He like said
And that's where they got it.
He runs our.
Like literally, actually, I was like my group leader. And I just made like, I was just like, oh yeah, yeah. Like I made it with my friends on Facebook and made like a chat. And like everyone just had it all done and I was just like,
What the walls are made of?
The lady.
Lightning
What's
is
she called, like the advisor or whatever.
oh, this is my turn. I don't know her name.
Ours is Jill, we did not talk nothing about, like.
Gram positive
none of that.
have a cell wall.
So, I'm gonna give them our.
Gram negative do not.
They
have
told
a
me,
cell
but she
wall.
said y'all could charge them like.
Everything has a
I don't
cell.
have Facebook for that.
Gram negative.
Gram positive
Text me,
have
text me.
It's
But they
just
are still right.
like it's negative has a cell.
We got the most friendly.
Yeah, if you go to the Good Sam, like the main college website, you just go to about us, and then click history, there's like a thing about,
I'm sorry, I didn't do anything, but I think I do all the work.
So you know how some people are like, I know I can be like this, and then you gotta make sure everybody did their part, or make sure I'm
You never see that, you didn't have to do
no, I want to like you for something. My grades.
Right.
Wow. I need to like that.
See, with me, I hear like I want to get it done, but since someone just took it away, like it was just nice. And it's just like, all right. Go ahead. Usually I'm
I don't even think of that. I don't even do something there. I'm just like, you know, I'll go run it for maybe
I'm down.
Okay.
Like, take down.
Yeah. Yeah, we have.
Shit, I was tired.
But,
Oh my gosh.
I had a fan on me last night. Don't lie. I was like, I got out of the shower and literally thought I was just gonna pass out, so I laid on my bed with like, my fan and I fell asleep. Usually I am with my dad.
I can turn it down to like
Our house is like our house is
Yeah, and I was like, what is going on? And then never mind. Yeah, I just kept punching it.
I mean, you know, you have council meeting for at the end of the start, like their age. Oh my god, right? It's like like pretty much every class. I And then I figured
That's what you drink is whole month. If they offer them out with those no matter. I mean, not even water. I didn't think that's gonna be Usually I'm the one that's like, I can't do this. Well, the first thing you He's hilarious. It was so funny.
calling over here. I saw you guys.
things down here that are strike Like this is the gram processor. It's got a thick cell. The gram negative's got a thin cell. This is the membranes right This one here is a covalent bond.
I'm thinking if they rock into
It was
Somebody asked me at break, it was a legitimate question, talking about the water thing again. Does it mean you shouldn't drink 8 glasses of water a day? No, that's not what
That's what you said.
Now, what was the point of the article? Not that,
The point of the article is it myth or not. And some things, they tell you to do, which you really don't have to.
okay, so there was no validity to the claims of drinking 8 glasses of water a day. Does it mean drinking 8 glasses is going to hurt you? No, it doesn't imply that at all. So if you like drinking, if you're thirsty, drink 8 glasses of water a day. But just that this myth, this kind of mystique that says that somehow if we do this, something, like vital spirits come out of the water and make us different. So it's, that's wacko thinking, so don't do that. Okay, next slide up for business here is different from the one we looked at before we broke. And let's look and see what's the same here. Well, we've got this thing down here, and of course that's a plasma membrane, cell membranes, cytoplasmic membrane, all names for the same thing. It's that biphospholipid membrane type of a thing. There actually are some variations between this and that, but let's not go there. Let's just talk about this is a plain old cell membrane. It's got its proteins stuck in there for transport for things that can't dissolve their way through that. We know that. But look at our peptidoglycan. So what do you notice here? Okay, so the peptidoglycan is maybe 1, maybe 2 layers in thickness. But there's something else weird about it. If you take a look at the handout that I was kind enough to give you, and look on the back side again, what do you notice? There's your GM. That's all the same, correct? So your sugars are the same, but what's different?
Between 2 layers
The cross bridges are irregular. And so when the cross bridges are irregular, what happens is, instead of the sugars lining up nice and neat, what do they start to do? They're not parallel anymore, and you end up with gaps in between them. So the peptidoglycan layer becomes irregular because of the fact that we don't have regular cross bridgings of these short chain amino acids. But then, what else? What's the rest of this crap up here? What does that look like on the outside? Yeah, it looks like another phospholipid layer to the outside. And it is. But it's different than the cytoplasmic membrane. But it's still a membrane-like structure. It's got these things hanging down, those fatty acid chains hanging down. It's got some kind of a glycerol molecule out here, modified in one form or another. But this is still, even though it's a membrane, and this is where a lot of students get confused, so make sure you're on board with me here, that this outer membrane is not a cytoplasmic or cell membrane, it's part of the cell wall. So with a gram-negative organism, we find a variation in the peptidoglycan. It's thin, irregular, but then it has to the outside, what? This outer membrane. But that outer membrane is still part of the cell, what we call the cell wall. Now, what's all this other stuff in there then? So, one of the things you know is that if this is going to be some type of a phospholipid layer out here, we're going to have a tough time getting material that doesn't dissolve in lipids through there. So what must be there? We've got to have some rather significant transport proteins or channel proteins or porins, call them what you will. But in turn, there are some other chemicals or chemical-like structures that are found here. And what I want you to focus on right now are these. Looks like hairs coming out. Those things, strand-like things. And these are what we refer to as LPSs, which is a shortened version of a lipopolysaccharide.
Lipo, you know what that means. Poly, you know what that means. And saccharide, referring to sugar. So it's a sugar with some lipids attached to it. And then we've got a variety of other interesting kind of support molecules and other transport molecules here. But I want you to keep in mind, in the back of your head, because we'll come back and review it later on when we get into medical microbiology, that these LPSs are an important virulence factor for gram-negative bacteria, because that's what this organism is. It has a cell wall like this. This is typical of gram-negative bacteria. And one of the important virulence factors, one of the important capabilities of producing disease associated with gram-negative organisms is going to be this lipopolysaccharide, this LPS, which is unique to gram-negative cell wall structure. Yeah?
So gram-negative, they're the ones more prone to becoming resistant?
No. Okay. No, we can find resistance in both types. Both types. What I want you to focus on right now is the difference in this cell wall structure. So we saw what gram-positive was. Thick peptidoglycan, well organized, always looking the same, bound together by these cross bridges that are very regular. No outer membrane. With gram-negative, thin, irregular peptidoglycan, but this outer membrane. Yeah?
So is the gram-negative more difficult for things to pass through because it has the membrane?
Exactly, because it's got this outer membrane, it's going to mean that it's got to have mechanisms whereby it can get water-soluble materials in and out of that. Now, one other thing to keep in mind here is, look here. This region right here. So what we have, and this is oftentimes just called periplasm there, it's called a periplastic space. Perimeter, goes around, plastic, fluid, space. It's a space around the cell membrane, which has a fluid. And that periplastic space, oftentimes, getting back to your point, can things travel easily in and out of that outer membrane? In some cases, no. And in many cases, a gram-negative bacteria will produce waste products that it kicks out, but they can't make it way through the outer membrane, and they get stored in this periplastic space. For that reason, I'll throw this out to you right now, and we'll talk about it more later on in the course. Do you always want to give an antimicrobial to a patient who's got a gram-negative infection that is cidal, meaning that the antibiotic or antimicrobial will kill off the cell, or do you want to use a static antimicrobial, which slows reproduction? So you need to know the extent of the infection and what's involved, because if you simply bombard a patient with cidal antimicrobials that will kill the cell off, all of a sudden you get all these toxins dumped and the patient dies because you gave them the antibiotic. You kill off the cells, all those toxins stored inside that periplastic space get dumped in there, the patient goes into anaphylactic shock and usually acute glomerulonephritis, and you kill them. So that's why sometimes it's a crapshoot to deal with a patient who's come in with a severe infection. What are we going to do here? Do we use the big guns and kill off those cells, or are we going to try to stabilize them by just slowing down the reproduction of those cells in the guy's body? And for that reason, when we talk about medicine, it's what? It's not medical science, it's medical arts. Meaning that it's what? Your best guess. Yep.
Where are the toxins stored? I'm sorry.
In this periplastic space.
Okay.
So that if I were to use an antimicrobial which destroyed the cell wall, I would dump all these toxins out. Everybody with me on that? So you got to know what to do. It's not the idea of here's an antibiotic came up, antibiotics cure everything. They'll
kill you if they're used incorrectly. Yeah.
I've seen that happen, where you can, with that same situation, my son came in and he had an infection in his leg.
Yep.
And they thought, at first they thought it was a necrotic fasciitis, but
Yep.
it wasn't. They dumped him with all these antibiotics in a 12-hour period, almost shut down his kidneys, and the infection just kept going because
Yep.
there was no correlation.
The best example I give, even though he didn't have a gram-negative infection, was Jim Henson.
Yeah.
Gurney,
Oh, yeah.
Gurney, you know. He had a strep infection, and he let it go way too long. And by the time they got him, I think it was Presbyterian in Manhattan is where he finally ended up. They didn't know how to treat him. And they went for the big guns. And what happened in that case is that he had such a severe infection that the killing of all of these cells put him into anaphylactic shock and severe glomerulonephritis, and the treatment killed him. So it's one of those cases, again, what do we do best? Was he going to die anyhow? The doctors were saying he was on death's door, so we did the best we could. But what they realized is that they shut his kidneys down with the treatment. Yeah.
What we found out is from the very first, I guess, swab or whatever, that it was gram-negative, and they used too many antibiotics.
Yeah. Happens all the time. Happens all the time. But again, it's medical arts, not medical science. Yeah.
Okay, so gram-negative, we know where it's being stored, like the junks in the periplastic.
Yeah.
Okay. Now, when you have gram-positive,
They don't store those toxins.
It's not because it doesn't have that second.
Right, it doesn't have that region around on the inside of the peptidoglycan. This is where this lies. This kind of whole, this area right in through here. And it's that outer membrane that holds that stuff in.
Okay. Right, because it can't get through.
Exactly,
Right.
exactly. Good point. Okay, yeah, go ahead.
If it's producing all this waste and can't get rid of it, how come that doesn't break it down?
You know, it gets rid of it slowly. It seeps out slowly, but it's still concentrated enough so that if we were to break that cell wall out there and just release these into the body, our cells can't deal with these toxins. It's a massive overload. Toxicity is what it amounts to. Yeah.
Is the peptidoglycan what stains well?
Okay, we're getting to that. Hold on. Okay, so here's a cell wall. We've seen gram-positive, gram-negative. There are some bacteria that don't produce a cell wall. One example of that I can give you would be this organism. Mycoplasma. I'm going to write SP out there. And let's talk about this real quickly. So when I start to use these words like this, what does that word mean? It implies what level of classification? And it's underlined. First letter is capitalized only. This is a genus name. So Staphylococcus would be a genus name. Streptococcus is a genus name. Mycoplasma is a genus name. Escherichia is a genus name, which people can't pronounce, so they call it E, right, for E. coli. So in the case of E. coli, E representing the genus name, space, coli represents what? Species name. And what do we call this? This is the correct scientific way of naming a cell. Genus name, species name. What's capitalized? Only thing that's capitalized is the first letter of the genus name. Everything else is lowercase, with a space in between. If you don't underline it, what else can you do that's appropriate? Italicize. Got it? You get it? Okay, so back over to this again, getting at this, talking about what's the scoop with Mycoplasma and species, because there are several species out there. One of them is pneumoniae. This is the organism that causes walking pneumonia. Does not produce a cell wall. Does not produce a cell wall. And I'll show you where problems lie with that in just a second here. So just to show you that there are always exceptions to the case. There are way too many bacterium out there, and it's not like everybody lines up and fits in these nice little boxes here, there, and whatever. Well, what's the function of the cell wall? Expand on that. Say it again? Okay, so it maintains morphology, maintains shape. Say it again? No. No. Maintains osmotic strength to the cell. Because what happens to a bacterium is that it typically gets moved from one environment to another, placed in different osmotic pressures, and what we see is that with the cell wall there, it allows for the cell to be in strongly hypertonic, hypotonic, remember all that stuff from AMP1? So that the cell doesn't burst. So it provides strength, you were getting at it there, but especially under harsh osmotic situations. The cell wall plays an important role. Okay, what does this have to do then with gram stain? And I'm going to briefly talk about that, but when we get into lab and actually do these, I'll go in much more depth. Okay, here's the scoop. You ready? Say sure. So, at the top, bacterial cells. Got a streptococcus down here, got a couple of different kinds of bacilli right there. You look at them under the microscope, you don't see anything. They just appear like little gray cells. We use something called crystal violet first, and all the cells get stained by this stuff called crystal violet. You'll be doing this dozens of times in the lab. So all the cells stain purple. We had iodine on there, which is a mordant, and we'll talk about that in the lecture. It just helps to solidify or engage the crystal violet within the peptidoglycan layer of the cell wall. And notice I use the term peptidoglycan. So where does the crystal violet go? Down into the peptidoglycan layer. Iodine there, and then we use alcohol. And when we use alcohol, which is sometimes called a decolorizing agent, some of the cells lose their crystal violet.
Would that be the gram-negative ones?
Hush. Some of the cells lose their crystal violet. And alcohol dissolves what component of a cell wall? Does it dissolve the protein? No. Does it dissolve the short chain amino acids? No. But what dissolves in alcohols? No. Lipids do. And what is the outer membrane of gram-negatives? Mostly lipids. So any crystal violet that got caught the purple stain gets lifted off. What do we know about the peptidoglycan in a gram-negative cell? Instead of it being lined up with all these stains stuck in it, it's got these gaps in it. So we easily lift off majority of the crystal violet. We counterstain with a red stain called safranin. So gram-positive bacteria stain purple because they retain the crystal violet. Gram-negatives lose most of the crystal violet and easily counterstain with a red stain or a pink stain called safranin. So what do we see? Gram-positive, purple, gram-negative, pink. So the colors, the variation in colors is due to the structure of the cell wall. Everybody with me? In lab, we'll talk about that in much more depth. But let's talk about one other thing as well. We were yakking about antimicrobials. Let's run with that for a second. Because we know that certain antimicrobials those that are narrow spectrum antimicrobials, antibiotics call you with, but there's a difference between those two terms. Some are narrow spectrum, which means they only work against gram positives, and some only work against gram negatives. So let's take an example of one. And I don't want to steal thunder from later on in the course, but this stuff. Penicillin, V or G, either one. Penicillin V, penicillin G, either one of those works effectively against what particular group of bacteria? Gram positive. Gram positive. And the question is, why does it work against gram positive organisms? And it works this way. Here's a bacterial cell. Let's just draw it up here. We're saying that it's a bacillus form. It's a rod-shaped bacteria. It's got this cell wall around the outside. We know what that looks like now because we've been yakking about it for an hour. And penicillin has absolutely no effect on this cell in this state. It's an adult cell, it's a mature cell, it's doing its thing, no effect from penicillin. This cell divides though, which we know they'll do, they'll reproduce, they'll divide. And when it divides, let me just move this one over. Here's the cell membrane. Obviously the membrane's going to pinch off. And what? Each of these cells is going to grow, correct? Down its full size again. But what must they add on? New cell wall here and here, correct? They have to build a new cell wall because the cell has divided its DNA. That went to either side, produced new cytoplasmic membrane. We split these across. But we had to make new cell wall here and here. Penicillin functions by interrupting those cross bridges of amino acids. So what happens to the cell wall?
It's too weak.
It's weak. It has none of these cross structures which link these chains of carbohydrates together. So what happens is that this turns out to be an exceptionally weak cell wall, and this cell gets placed in an osmotic situation. What happens? The cell dies. Keep in mind this, and people always want to know, and they're most critical, the patient is most critical with penicillin, because they want penicillin to work in 3 days. But penicillin works 8 to 10 before you notice the effects in terms of change of infection. And what's the reason for that? You've only got less than 10% of the cells reproducing at any one given time within a 24-hour period. So giving penicillin, how many of the cells have you killed off? This many of all of those that are causing you the disease. So the next day you're going to kill off another chunk, and with lab studies we know it's going to take 8 to 10 days before you kill off enough of those that your bone immune system kicks in and you deal with the disease. But what do most people do? Third day they call their doctor and go, this stuff doesn't work, give me something else. And what do doctors do typically? What do you want? Although they're getting better with that, but for a while there it was, what do you want? You know, I don't have time to deal with you whining and crying on the phone telling me the stuff I gave you doesn't work. And do they have time to educate you? We need to talk about cell walls. You know, and considering how many people in the world are uneducated, I mean, this is foreign language. So that's why you need to take the full regimen. And what's the worst part is, of these cells that are dying, they're the weakest of the lot. Who keeps surviving every time you give? Who keeps surviving? The strongest, those that are the most resistant. And therefore, if you again don't go the whole regimen, what you've left is this whole battery of super strength microbes in your body that are going to beat the hell out of you. And then you come down with a super infection after that. You can't figure out why. That doctor gave me the wrong medicine. I've heard it 1,000 times. Okay. Going back to mycoplasma. Mycoplasma, how many of you had walking pneumonia? more than I think probably know you've had it. In some cases it's very mild. You get a flu-like or a cough-like syndrome, weakness, and so on and so on. You typically think, what do we think? We get a little sick, what do we always say? Cold. No, you say I got some flu bug. Okay? But in this case, does penicillin work? If it produces pneumonia-like symptoms, which would be typically associated with certain strep or staph organisms, which are gram-positive, and you got a physician that doesn't read the symptoms right, and he or she gives you penicillin, why doesn't it work? No cell wall. Right? So now you've got this penicillin V or G you're getting, and you wonder why you don't get better. Well, how does it work? It interrupts cell wall formation. These don't have cell walls. Absolutely ineffective. So again, I hope what you learn out of that is that what? I've got this vial of antibiotics sitting in my medicine cabinet at home. I have no idea, I can't read what it says on there, but it's an antibiotic, it's got to cure everything, right? And loads of people do that. They hoard their meds or steal them from a neighbor and just waiting for an infection to come along so they don't have to make a trip. Okay, moving along. What else can we talk about these prokaryotic organisms? Most of them have cell walls. If they have cell walls, they vary into one form or another. We said before that bacteria have ribosomes. And what you can hopefully remember from your A&P days is that a ribosome is actually made up of what two substructures? Ribosomal RNA and
Wait, okay, is
Okay.
that like mRNA and no?
No, this is ribosomal RNA. There are different types. There's so many different kinds of RNA. We're used to protein synthesis. We think of messenger RNA and transfer RNA and all that. Well, ribosomes have something called ribosomal RNA along with a protein. And if we take a look here, this is kind of, and I'm not asking you to memorize this, so hold on, don't get all bent out of shape. So here's a typical ribosome, a eukaryotic ribosome like in our cells. And what I want you to notice is the difference between these two. If I centrifuge these things out, they centrifuge out at a certain rate, so on and so on down here. Anyway, the point of the whole thing is that I get 80 Sverdrup units on a ribosome for a eukaryote. Simply means that compared to a prokaryote, which has 70, these are slightly different. Ribosomes are slightly different. They both do the same thing, site of protein synthesis, correct? Just say yes. Somebody say amen. There we go. So, okay, what's the difference? Well, since we've been talking about antimicrobials here now, and antimicrobials work in different ways, and what was Ehrlich's point working with Salverson trying to treat syphilis? Magic bullet. Give me a chemical I can put in my body that only goes after the organism, doesn't go after my cells. So one thing we've seen so far is we got a whole bunch of antimicrobials that can work on cell walls, because are there any cells in your body that have cell walls? If you say yes, I'll kill you. The answer is no. We don't have cell walls at all. So that's why penicillin works, right? It tends not to affect us, except that it's still a foreign chemical and some people are allergic to it, right? Why not? It's foreign chemical. Your body goes, what is this stuff? And some people go overboard, and other people go, I can live with it. So now, think that logic, the Ehrlich logic, that what? If I want to kill off what's causing me the disease, I got to find what's different about that cell. And now, we have a whole series of antimicrobials which do what? Can latch onto the ribosome. They don't latch onto the ribosomes in our cells because they're two different shapes. We make one that just fits this one. What does it do then to the bacteria causing the disease? Keeps it from producing protein. And as soon as you start to lose enzyme systems for metabolic regions, you die. Make sense? Make sense? Say yes.
Yes.
So how many of you are lying?
Yeah, I am.
Okay, good. So you want to go through it again? Want to walk through it again?
Yes.
The important thing I'm saying is that do our cells have ribosomes? Do bacterial cells have ribosomes? The answer to that is yes. Do they do the same thing? Yes. It's the site for protein synthesis. On the other hand, they are slightly different from a molecular point of view. They're slightly different from one another. So if I want to use an antimicrobial which will kill off bacteria, and I can take that chemical, it makes its way through the body, gets inside the bacterial cell and latches onto, it fits this ribosome, but it doesn't fit this one, which is ours, but it will fit this one. What does it essentially do? It stops protein synthesis in the bacteria.
Okay.
Okay.
I get it.
There you go. Sometimes it just takes a couple of times, doesn't it?
Right.
And you should always ask. Always ask. Yes.
So by attaching to the protein with the bacteria, it's killing the bacteria, not the cell.
But it won't kill us.
But it won't kill us.
Yeah, so it's that magic bullet again. So we got to come up with some type of an antimicrobial which just fits ribosomes. But which ribosomes? Bacterial ribosomes, those that are of a 70 Sverdrup unit. And I'm not asking you to memorize this Sverdrup unit stuff.
Do all bacteria have the same ribosome?
Yes. But never say yes and never say never.
Unless you tell us to say yes.
Exactly, exactly. Okay, so we've got ribosomes we could deal with. Again, what we're talking about here, what do we find inside prokaryotic cells or associated with prokaryotic cells? Well, one of the other things we see is that from time to time, there are certain types of bacteria that can produce these things called spores or endospores. And this is a term that's going to confuse the hairy out of you in this course. The use of the term spore. Because when we get into the fungi and start talking about them, we're going to use the term spore there. We'll see with, the term is used with certain protists, and it gets really confusing. But in this particular case, and again, I'm not asking you to memorize all the steps. I'm going to walk through them with you and show you the end result, and that's what I want you to focus on. Some bacteria, not all, mostly gram-positive bacilli, gram-positive rods, those kinds of bacteria are the ones most prone to make spores or endospores. And how do they do it and why? More important is, why do they do this? Well, when environmental conditions get bad, and the question should be then, where do we find most gram-positive bacilli? And they're found in the soil. They're soil organisms, they tend to be. So when you were a kid out there playing in the dirt, and you got all these bacteria because you were scraping and digging around there or doing whatever you're doing, and then you pick your teeth or you clean your nose. You picked up billions upon billions of these organisms. They never caused you disease, but they stimulated your immune system, and you're a better person for having done that. Environmental conditions get bad for these bugs, these soil organisms, and some others. And what do? They say, well, either I'm going to die here, there's not enough water, it's too hot, it's too cold. Usually they don't die when they're too cold, but they can die if it's too hot. Toxins in the soil. That bacterium over there is producing an antimicrobial that's going to kill me if I don't have the ability to get out of the way, so I'm going to somehow save myself, and I'm going to produce this endospore. So we start with a vegetative cell, and in turn, what happens with that is that it's separate, replicates its DNA, moves to the part, and as it allows then the new DNA to start to have these layers built around the outside the other DNA crumbles and disintegrates. We produce this kind of shell around this DNA. The cytoplasm is gone, and we're ending up then with a spore, a free spore. It started off as an endospore inside the mother cell, ends up as a free spore. It simply gets dumped into the soil. Is it a means of reproduction? No, because one cell gives you one cell. So it's not reproduction, it's simply survival. These endospores, or these free spores, can last indefinitely. You know, they open up a tomb in Egypt that hasn't been unsealed in 6,000 years. They'll find spores on the linens, on the mummy, and they'll grow them out again in plates to give them the right amount of water, and here comes the bug right back again.
So how does it reproduce?
It's not reproducing.
I mean, so how does it spread? Like, how does it, what's its?
It doesn't go anywhere unless it gets carried away by wind or water. So this cell, in this place in the soil, now environmental conditions are bad. It knows it's going to die, so it's going to protect itself, and survival is the key. So it goes through a process where it produces a spore, no cytoplasm, just the DNA, and this multi-coated kind of a structure around the outside, and it's a protective kernel, which we call a spore.
So no, like antimicrobial or anything can get to
No,
that?
no. Very difficult to kill. This is one of the reasons why. Now, for instance, let's draw a scenario. We have a severe rain, and you live out in Brown County, and the water that, which I don't know if it's the case, but let me just draw this scenario. The water that you use in Brown County comes from retreated sewage water, and you end up with a heavy-duty rain, and the water treatment plant can't keep up with the influx of water coming in, and there's a cross-contamination between the sewage and the cleaned water which is going to your house. And they tell you then at that point to do what? Boil for a minimum of 15 minutes. A minimum of 15 minutes. What are they getting at? Endospores. From the soil, they got washed into that. So if you boil for a good 15 minutes minimum, you can probably kill off most of the spores. But a quick 2-minute, 3-minute boil, they laugh at that. Yes?
So it doesn't start off as one endospore, it starts off as a bunch of endospores.
No, it starts off as a cell, just a regular bacterial cell. He's cooking away in the soil, doing his thing. Conditions get bad. You know, it's one of those summers where the temperature goes up to 110 and stays that way for 3 days. He's living just an inch below the soil. It gets so hot, he's going, I'm going to die unless I save myself or herself. And so this one cell produces this, what we call an endospore. The mother cell dies, and we're left with this, what? It's nothing more than really a multi-layer of sugars, insoluble sugars, with the DNA of the cell inside. That's all it is.
So to get multiple spores, you have to have multiple endospores.
One cell gives rise to one spore.
So is it kind of like a peel bug that rolls in a ball?
No, that's a bad analogy.
And there's
No what?
what did you say there, the endospore has no, cytoplasm?
No cytoplasm. So it's in suspended animation.
Yeah.
It's not doing anything. It's not metabolizing. It's not using up anything. It is simply protecting its DNA and waiting till conditions get right again.
So how does it come back to life? Is it like a gremlin? Does it need water? Like, I don't understand.
Vital spirits.
I mean, but for real.
No, what happens is that the water comes back, gets it back into an aqueous environment. It can start to absorb perhaps even some carbohydrates from the environment, dead leaves and things like that, and they slowly make their way in. The cell, it reactivates the chromosome, and we start then to say, okay, I've got enough water here, let's start to make some cytoplasm, let's start to digest away this kind of shell we build around our cells and rebuild a new cell wall. I'm going to make new ribosomes and it goes back into a vegetative cell again. Okay? Okay. Do all bacteria produce spores? No. We find most typical what? Gram-positive bacilli, that are rods that live in the soil. By far the most common.
Wouldn't it need to save some ribosomes as well?
Say it again.
Wouldn't it need to save some ribosomes as well in the spore to be able to reproduce?
That's a good question, and it seems that some do save a few of those because they need to kind of get this enzyme system going up again. So it would seem that there would have to be some of those that got stored.
And DNA by itself can't reproduce.
It needs the enzymes to do that. Exactly right. That's a good point. Some of these prokaryotic organisms, these bacteria, are motile. And they're motile in a variety of ways. But they have a flagellum for those that are flagellated. So you see we got a cell here with these little things kind of sticking out of them. And there are other means of motility in bacterium as well, but for those that are motile by flagellum, we've got a unique kind of a situation here. When you think of flagellum, what comes to mind in terms of movement? Would you be willing to stand on the table and do that again?
No, because it has little things coming off of it, so I just thought I don't know. It's the noise the flagellum makes.
Okay, I'll move down here. When you think of flagellum, think of sperm, and what's the action, right? It's the only flagellated cell in the human body. It's whiplash, right?
Yeah.
Okay, so it's a whiplash motion. Propels that little guy along quite well, does really well. We've got modified flagellum called cilia that are not involved with movement of cells, but movement of material, right? So you've got ciliary escalator in your trachea. You know that, right? Its purpose is to move mucus up to your throat so you can swallow that stuff. But when we even think of cilia, what do? They've got this kind of motion. Well, if we take a look at bacterial flagellum, and for only those species that are flagellated by these, this tends to be the form. There is a rod which runs down through, and in this particular case it would be typical of what kind of, it's got an outer membrane, what kind of bacteria? Gram-negative. And this rod runs down through that. It's got some discs that anchor it into the outer membrane and the peptidoglycan, and then it has another set of discs that are anchored in the cell membrane. If it's gram-negative, it's missing these discs out here, quite obviously. So you got this rod which can spin inside these discs. And to that there is a hook-like structure, and then we end up with this filament that runs off, made up of these subunits of protein. So it's a hook-like thing, and so what we end up with is motion like this. Not like this, I'm saying, not like this, like with sperm, but this way. So it spins on this rod. It functions basically then as a propeller-like structure. And if we use that, how much time we got here? We'll end on this one. Let's talk about this. If you think about that then, so here's your cell, and maybe that flagellum will come out. Here's the hook. It's got the discs down in here and the rod coming through, and it's got the flagellum that's coming out there. That's pretty easy to understand how that would work, because it acts mostly like a boat propeller, right? Spins. And the thing is that this then could allow this cell to go which way? That way or that way. If we find a cell which has a single flagellum like that, we say that it possesses monotrichous flagellation. Monotrichous flagellation. Mono simply meaning one. Mono. Trichous comes from trichome. Ring a bell for anybody? What's a trichome? It refers to a thread-like structure or a hair-like structure. So we've got what? One hair-like structure coming off, one flagellum. And it's pretty easy to understand how an organism could move back or forth if it had one of those. Spins one way or spins the other way, moves back and forth. In turn, there are some bacteria that have not just one at that point, but have numerous at that point, a tuft of flagellum, as it were, at one end of the cell. And we refer to that by a couple of names. Typically, Lophotrichous flagellation. Let's forget the mono because that'll be confusing here. Let's just use the word Lophotrichous, L-O-P-H-O. Lopho, which refers to a tuft. Again, the point that I was trying to make out of that is that you can have Lophotrichous flagellation at one end of the cell. You can have monotrichous flagellation at one end of the cell. Or what could we have? Monotrichous flagellation at both ends of the cell. What might we call that? It's called amphi, mono, or Lophotrichous flagellation. Amphi meaning at both ends. So we could have one flagellum at both ends, or we could have what at both ends? A tuft at both ends. And I could go on, but I'll give you just one more, because it gets kind of crazy with this. Or we've got some bacteria that have these flagellum sticking out all over the place. And that's referred to as peritrichous flagellation. Simply means you got flagellum all over the place, all around the cell. Yes, my dear.
Okay.
This better be good.
So, you said it turns in a circle like a...
It spins.
It spins.
Yes.
Okay.
Do you see
But-
some problems?
Yeah. All over the place. It'll
Ex-
want to stay in one spot or something.
Yeah. How does it then move? How is it directional? Is the question. So if we've got one maybe at one end and one at the other, I could deal with that. One spins one direction, the other's going to counterspin, and we can move in that direction. But what happens if I've got a tuft of them? So envision this. Envision this, that you got legs coming out all over the place on your body. You don't just have two, you've got 9 of them. And how many of you find yourself tripping going up and down stairs? With 2. Okay, so we got a fair amount of you. Now try and do that with 9. Trying to coordinate 9 legs going up and down stairs at the same time. And you can understand the difficulty. So how do they get around this? Put your hand down, because I'm on a roll, and I'm going to tell you. What they'll do with Lophotrichous, this cluster, is that they will do what looks like a French braid. They'll wind them all together, and then they function as a single flagellum. Spinning clockwise, and in turn, they can spin counterclockwise. Now, what gets even more interesting is, and you can see if they were Amphilophotrichous, you would want to have one spinning one direction at one end, grouped together down here and spinning the other direction, so you get unidirectional movement of the bacterium. But what happens if you're peritrichously flagellated? What's got to happen?
Some are on and some are
Now you may go.
It has to hook on to something?
Nope.
No, I'm sorry.
They somehow got to all be coordinated. So now you go, wait a minute, this is a little single cell. You're telling me that this cell is capable of reading its environment, it needs to move. Why is it moving to begin with? And it controls the direction it's going to move. So it's sensing its environment, and it's saying, I'm going to have my flagellum spin in this way to either go toward that or away from that. Simple cells, huh? And we don't have all the answers to that in terms of how they're capable of sensing these environmental stimuli. But what might those environmental stimuli be? Well, some of the things we just talked about. Toxins over here in the soil, too hot, no nutrients there, nutrients over here. I'll move over here, more moisture over here, I can move over there. Depends on where we want to go. It's capable of sensing its environment and then in turn moving. And what's fascinating about bacteria, and you can see this under the microscope, it's the neatest thing, if you have the setup right, is that you start here, and you can take a well slide, and you can put a drop of bacterium here, and over at the other end of the well slide, you can put a small, very loose concentrator, not loose, a very small concentration of sugars like glucose. So we got food over here. You put a drop of bacterium, modal bacterium in here, and they will go that direction, and they do a tumble. This is called a run, R-U-N, regular run. They'll do a tumble, they'll go another run, they'll do a tumble, they'll do a run and a tumble, and another run until they're where they want to be. Well, what's going on there? What are they doing with every tumble? It's not like they're lost.
Redirecting towards them.
Exactly. They're looking for the best stimulus again to say, this is where my next run is going to be. And with the bacterium under the scope, you change this halfway through, and they will move in that direction and do a tumble and get to that run. Again, what are they capable of doing? Sensing the environment, and in turn, being able to move back or forth towards something or away from something.
This is peritrichous or?
This is any flagellation. The question is, why would they be modal? We don't care how they're modal, whether it's uni or monotrichous, whether it's lophotrichous, whether it's peritrichous. Why would they be modal? And the answer is to do one of several things. They're reacting to environmental signals. I want to move toward that, or I want to get away from that. Okay, enough for today. Lab people, today 1:30. We'll talk about and see if we can move that up to 1 when we group together. Same as you people on Tuesday, but we'll talk about that as a lab group when we get in there. But so for today it's 1:30, and it's fun.
Turns in a circle, right?
Yeah.
So why do they make the whiplash type? Usually the side to side is still in a circle.
It simply acts like a road propeller, and it can coordinate all of its function in the same kind of a way. So they're all coordinated activities when we bounce them. So how does that single cell know how to do that?
Don't pay attention to something else. I mean, if they have,
We'll get to attachment next time. We'll talk about
I think when I was looking at the picture, they were in the mono.
Don't get Hold on, we'll talk about attachment next time we get together.
I think this is another slide up there. I have that one. Is it
I'll
still
check
in
over
this?
there.